Assalamu alaikum wa rahmatullahi wa barakatuh. Mr. Abdullah Amin is talking, and welcome to our biology lesson. And today, inshallah, we are going to explain chapter 8.5, the second part of translation. In this part, inshallah, we are going to know what is anticodon and the protein factory, which is ribosomes, and how translation process happen. Okay. Anticodon, guys. Anticodon is a set of three nucleotides. So we have three nucleotides that that are complementary to the strand of the messenger RNA. So these three nucleotides are complementary to the messenger RNA, which is a codon that found in the messenger RNA. We know that the codon, which is like a series of or three nucleotides beside each other that can represent a certain protein. So the anticodon is found here in a structure, which is this is transfer RNA that carry certain amino acid. Okay, so three nucleotides complementary to the nucleotides that found in the messenger RNA and carried by transfer RNA. We know that, guys, the protein is made up of chains of amino acids together. So when these amino acids become in orders or like it's grouped together, they will form a protein inside the place where the protein made, which is ribosome. So let's talk about the ribosome. Ribosome, guys, has like made up of two subunits. One large subunit, which is like make the binding site for the transfer RNA, and this small subunit, it's responsible for binding to the messenger RNA. Okay, so this is all of it, which is the structure of ribosome. Okay, let's see, guys, how a protein is made inside this ribosome. We have, guys, this messenger RNA, which has come out of the nucleus after transcription. So it's go to the site of ribosome in the cytoplasm. And as we know that from the last lesson, we have something called the start codon. Okay, so a start codon, guys, which is like can attract the transfer RNA, so to signal the ribosome also to continue this process. And then this exposed codon can attract complementary transfer RNA to join to the messenger RNA, and then the amino acid, it will be like assembled together by this way. So, guys, the exposed codon in the first site that can attract complementary transfer RNA that have the amino acid methionine, and we said this is the start codon, okay. And then, guys, this also exposed codon, like it can also attract another transfer RNA with another amino acid to join to this site, and so on. One thing else, guys, the ribosome can help to make like a bond between these amino acid. This bond is called polypeptide bond. Okay, and then the ribosome, it will move step by step, codon by codon, length of one codon at a time. Then, guys, this empty transfer RNA molecule can exit the ribosome. And here, guys, the exposed codon, like it can attract transfer RNA with a different amino acid to bind to the previous amino acids, and so on. Once a lot amino acid, a lot of amino acid are binding together with a polypeptide bond, so we have a long chain of amino acid together. And finally, guys, once the stop codon like is reached, so the ribosome can release the protein and disassemble. Okay, let's watch, guys, a video about this part to make it easier. Here, guys, how the DNA can transfer to protein. And this is a cell. And guys, we know that the DNA is enclosed in the eukaryotic cell inside the nucleus. Okay, so if you go inside the nucleus, you can find the DNA is like in the form of the chromosome. and each chromosome is made up of like condensed DNA wrapped around certain protein, which is histone. Okay, and then guys, each part of the DNA which is responsible for certain protein to make this part, which is called gene. To start making this protein, guys, we have to take a copy of this gene or of this part. So we have enzyme which is called RNA polymerase that like unwinding or unzipping the double helix of the DNA, and then nucleotides are added to make complementary nucleotides are added to make another strand of messenger RNA. Okay, so this is a messenger RNA. Before guys, like it's being used, it have to be processed like by adding or removing some sections of RNA, and then the RNA is ready to go out of the nucleus and go to the place where the proteins are made. So it leaves the nucleus and go to the ribosomes. Okay, we know that guys, this ribosome is made up of like two units, the largest one and the small unit. And another type, guys, of RNA, which is transfer RNA that can be attached to the amino acids according to the code that found in the anticodon. Okay, so this is transfer RNA that has anticodon, which is made up of 3 nucleotides. It can be, guys, bind to specific site on the messenger RNA, as we can see. And then, guys, the ribosome can make like bond between this amino acid, which is polypeptide bond. Then a lot of amino acids are added till the ending codon, and then this, guys, like a protein or chains of amino acids are added and the protein is made. Now guys, we finished the lesson. I hope you enjoyed this lesson and see you next time.